Okay, so we're about to start the next session. Next session is the keynote talk for the SIM4, computational biology and bioinformatics, system biology, and the modeling methodologies. So again, because we have a large auditorium, I encourage everyone to move front, so we can actually have a more close interaction probably during the Q&A session. Okay, so today, with a distinguished pleasure, I want to introduce Professor Shankar Subramaniam, who has been well known in the bioinformatics and computational system biology field for many years, to give the distinguished keynote lecture. So Professor Subramaniam currently is the department chair of bioengineer department at UC San Diego. He's also an inaugural chair professor in the department. Before his distinguished career in UCSD, he has also another very distinguished, successful career at the University of Illinois at Chicago, and Urbana-Champaign. He actually serving as the director for the bioinformatics program of the National Supercomputing Center. And also, he has been a strong advocate and a fierce leader in system biology and bioinformatics. I first met Professor Subramaniam 10 years ago because he was leading this international effort on Lipid map, which is actually multi-nation, multi-institution program. So that's why from today's talk, you would have a chance to see his distinguished accomplishment in how he applied the system principles to solve the biomedical problems. Such example including the famous biological workbench, which is a web-based tool that's being available to the wide community in biology and medicine. In addition to that, he has been recently started working a lot on understanding and uncovering the basic mechanisms related to the human diseases. So one is related to insulin resistance. What's the mechanism behind it? Use the high throughput computational system biology approach. Professor Subramaniam is a fellow of American Institute of Medical and Biological Engineering. He also received various awards from all the international organizations. So with that, I pointed out, again, you will really see what I say when I said that he's really a fierce leader in pushing bioinformatics and system biology through his talk today. So with that, I want to welcome Professor Subramaniam to give this talk.
Good evening, everyone. I would like to thank the organizers for this very distinct honor of speaking to the distinguished crowd here on engineering and medicine and biology. And I would like to, since I have the presentation right in front of me, how do I turn it on? Okay, I would like to begin with acknowledging my collaborators and colleagues, since it's, I almost never end my talks because they are infinite in length usually, so therefore I try to begin by acknowledging people who contributed to various things. So I'd like to begin with that. On the left two panels are people who have worked with me in the past 2 decades. They are past students and graduate students and postdocs. And on the right-hand side panel are people who are working currently with me, either graduate students or postdocs or scientists in my laboratory. We've been very fortunate over there. I've been really fortunate to have such wonderful colleagues and contributors to research programs that we have developed. And I also have been fortunate to get funding from a large number of agencies over the past 2 decades. We've had funding from the National Science Foundation, National Institutes of Health, which includes NIGMS, NIDDK, NHLBI, NIAID, NCI, State of California, Pfizer, Eli Lilly and Company, Mitochore Corporation, Silicon Graphics, Sun Microsystems, and 2 different foundations, Keck and Hillblom Foundation. So this is what makes the research possible. As I go along, I will tell you specifically about people who contributed to various aspects of research. I want to begin by talking about the role of engineering, the talk is going to focus on various aspects of examples from my laboratory which are showing the power of engineering in modern biology and medicine. I would like to begin by talking about the 3 basic pillars of engineering in measurement and analysis, modeling, and design. You can ask the question, has measurement not been done for a long time in biology and medicine? Yes, it's been done for a long time. But the high throughput technologies that have come forth in the last decade or 2 decades have really revolutionized biology. All you need to look at is the next gen sequencing, high throughput sequencing that brings up, that has the ability to sequence the genomes and epigenomes of humans. And so the analysis as a consequence becomes very complex because you have multiple scales, multiple levels of measurements, and how to integrate these measurements becomes a complex problem. So I'll focus on methods for measurement, methods for analysis to illustrate the power of engineering in biology and medicine. The second aspect, which is often ignored, is the power of modeling. When a biologist thinks about a problem, they also have a conceptual model in mind. But what makes it different in modern biology and medicine with engineering approaches is 2 things. One is the ability to do quantitative modeling, and the second is the ability to bring in vast amounts of knowledge to build in models at multiple scales of resolution. And so these are very distinct, and this is what makes engineering a powerful tool in medicine today. The last aspect of it, which is actually the future of where we are going for the next 2 decades, deals with the issue of design. Design is the foundation of engineering. However, in biological systems, we are still in the process of learning the precepts and principles which will go on to help us design systems which will both interrogate our physiology as well as be able to therapeutically solve various problems associated with pathologies. I'll give you a few examples of all of these pillars in medicine and biology. I want to begin with a slide which is from Peter Hunter. Peter is very well known. He's one of the awardees this year in the society. And Peter talks about this multiple scales in a living system, starting from genes to proteins to cell structure to tissue function to organ structure to clinical medicine. This focus of multiple scales that we can scan goes from, as you can see, from genes which are 25,000 in number to 100,000 proteins to 300 plus cell types to 4 tissue types to 12 organ systems to thousands of phenotypes. When we look at clinical phenotypes, day-to-day existence, there are thousands of phenotypes. So the integration of all of these diverse types of data towards building quantitative models is the challenge that I'm going to illustrate to you today. I want to continue with one more slide from Peter, which talks about the challenges in dealing with spatial and temporal scales. The difference between modeling in other systems and modeling in biological systems comes from this vast diversity of scales. You can start with anywhere from 1 meter per person, 1 millimeter per tissue, to about 1 micrometer per sarcomere spacing, to 1 nanometer per membrane protein. These are the rough orders of magnitude. If you look at time, it scans all the way from 10 to the 9 seconds, which is a human lifetime, up to 1 microsecond, which is the motion of molecules, which deals with the Brownian motion. So you can see we have multiple space and time scales. The first complexity deals with making measurements which are precise and accurate across all these scales. The second deals with creation of model systems which will help us probe these multiple time scales and multiple length scales in meaningful manner. In fact, the diversity of experimental models, Krishna talked about it before, using monkeys, primates as models. In fact, the multiplicity of models include everything from bacteria, which are, one of the early precursors of human life, of Earth life, and murine models and large animal models and human as experimental systems. And most importantly, we require a hierarchy of related models. In fact, this is really becoming the challenge of modern biology. We have data-driven models, which are driven by the vastness of data that comes forth, and equation-driven models, which are typical physicists' approach towards modeling. How to integrate these 2 things is really becoming one of the largest challenges in modern biology and medicine. And engineering plays a big role in terms of providing approaches towards integrating data-driven models and equation-driven models. I point here to approaches to dealing with gene networks, to pathway models, to statistical models, and I'll point out specifically how do you convert statistical models into equation-driven models. And these in turn are cast in the form of either matrix equations or differential equations, which will provide more quantitative framework for looking at input-response relationship. I'm going to tell you examples from my laboratory which deal with these things. I want to start by telling you the challenges that we deal with in systems biology. During the middle of my talk, I'll tell you about the challenges we face in systems medicine. In systems biology, the first question that comes about is how do we define and characterize a biological system? And this is half solving the problem. How can we obtain information on components of the system? We want not only qualitative information, but we want quantitative information. We want dynamical information. And these technologies and measurements have to have the ability to do this in real time. In fact, the previous talk by Krishna Shenoy told you about how you can do temporal measurements in the human neuronal cortical system. So I'll tell you more about the molecular and cellular systems, and then we'll talk about human physiology. What mechanisms can we infer from systems behavior? If you build these models from data, models from measurements, what type of phenotypes can we infer from these systems behavior? What are realistic, context-specific models of the system? In fact, again, I want to refer to Shenoy's talk where he talked about the fact that our conventional wisdom and view of what a motor cortex does is not completely correct because it's a system that you have to deal with input from multiple sources. So I'll tell you about context specificity as being an important component of modeling. How can we measure and compute input phenotype characteristics? How will we validate the model system experimentally? What do we use humans as models? Do we use animal models? This is very important that building models means you validate this is what engineers do and you redesign systems to help you understand the principles under which the system is built. And the problem with biology is that we have multiple dimensions. The complexity is very high because you have all these dimensions. If you talk about current ways of looking at things, people project multiple dimensions into 2 dimensions or 1 dimension and do analysis. And most of our models are built on simple 1 or 2 dimensional approaches. The complexity is in terms of integrating all these different dimensions towards understanding the system behavior as a whole. I want to take you through about 3 examples which deal with systems biology and a couple of examples that deal with systems medicine from my laboratory which deal with building technologies which will help you understand context, specifically what we are dealing with. B, I want to deal with pathologies and how we can interfere with pathologies using systems level models. First, I want to talk to you about spatial and developmental context in the formation of primitive streak and germline formation in human embryo. So if you look at conventional, many of you may know this picture quite well. If you look at the formation of a primitive streak, and in fact, it's the initial formation deals with splitting into different germ layers, which give rise to different lineages. It gives you cardiac lineage, neural lineage, etc. The question has always been, embryologists have worked hard on trying to understand precisely what contributes to this lineage specification, what contributes to germ layer definitions, and so forth. Conventional wisdom has been people have probed it at the level of one single molecule, and probing at one level of one single molecule, for example, people know specific transcription factors. Beautiful work from, for example, from Wisconsin has shown that you can take 4 transcription factors and force it to induce into specific lineages and so forth. The question then becomes, how do we map the complete parts list to build a model that will help us understand this? When we undertook this project initially, this was done in collaboration with Mark Mercola and the Burnham Institute. We had the following 2 problems. The first was an engineering problem, which is you can never get millions of cells from which you can extract RNA to do this. You only get a few cells. Think about the early lineages. You only get a few cells from which you can get RNA. The second problem is you have few genes that are expressed so highly that you're not ever going to see genes which are expressed in low copy numbers. So you're going to have a problem of dealing with this. So we built a technology which overcomes this problem. In this, let me first pose the biological question. The biological question we asked was, can we have a model system where we can gradually increase the lineage germline specification by activating using a chemical perturbation? And this is well known from before. This was not work done by us. This is work done by others, where it's well known by activating a TGF-beta ligand, you can force it to go towards a specific lineage. But how do we understand for each of these each of these stages of germ layer differentiation, each of these regions in your embryo, how do we know what are the parts lists that define the specification? This is a question that we posed biologically. To answer this question, we built a technology. The idea behind the technology is very simple. If you take conventional next generation sequencing, you take random primers, you take a large amount of RNA, you take these primers, amplify them, you sequence them, you align them to the known transcriptome, you say these are the genes that are being expressed at different stages. This is a standard technology. Illumina and ABI and many companies have pioneered these technologies which people use routinely. The problem, as I pointed out, is you need large amounts of starting material of these cells. You also need to look at only high expressed genes. So in our technology, we used an informatics strategy to take every gene in the whole transcriptome, identify unique regions of the transcript. We designed heptamer primers using an algorithm, suffix tree based algorithm, which helped us to build a complete primer list, a 44 primer list for the entire transcriptome. And this entire transcriptome expression now can be done using the standard technologies. And this will give us a lineage specification. So using this, we validated our method. You can see from the bottom diagram on the middle that we have got extraordinarily good agreement with known experimental results, but now we can specify the entire parts list for the different germ layers. So if you go to the next slide, we actually showed systematically how you can show with a model of this data. From this data, we built a model in terms of how do you show lineage specification in going from one germ layer to next germ layer to next germ layer. In fact, we mapped it to the entire embryo. You can see in the middle of the next slide, in the embryo, you can see the presence of the parts list in different regions of the embryo. And we now can build quantitative models in terms of increased expression, reduced expression, in terms of lineage specification. So we are pursuing this work in terms of specifying cardiomyocytes, in terms of looking at cardiomyocyte regeneration, so that you can have, ex vivo generation of these cardiomyocytes. This technology can be applied to a large number of other platforms. I just wanted to illustrate to you that bringing in novel technology in combination with the interesting biological problem has the potential to offer insights into how your embryo makes different germ layers. I want to go on to the next example. I want to walk you through a series of examples. We were very fortunate to be funded heavily by NIH to investigate one cell line in fantastic amount of detail. Over the last 15 years, we probably have the largest amount of data on a cell, how it responds to different stimulus. This is this first cell in the immune system defense. These are macrophage cells. We built the most exhaustive, let me show you in the next slide, analysis, most exhaustive measurements on these cells through 2 large glue grants. In fact, we built the perturbations on these cells. We included multiple ligands, 33 or 40 different ligands. We did expression profiling. We did mass spectrometric measurements. We did protein measurements, cytokine measurements. This is building the complete parts list for this large cell type. Using this parts list, we used reverse engineering approaches towards reconstructing networks. We can use a multiplicity of approaches. The very first thing, this is where data-driven modeling comes in contact with equation-driven modeling. We used a combination of statistical methods, simulation methods, optimization methods, regression methods, and clustering to build complex networks. I'll show you results of a few examples of these networks on macrophages. In the first example, I want to show you, we can take multiple ligands, multiple stimuli, and the macrophages are cytokine factories, so they produce inflammatory factors. We then know a part of the measured pathways. We know the unknown pathways. Our goal was to reconstruct this network so we can describe the input-output relationship in a quantitative manner. So we used the technology to statistical methodology, first to identify what is the crosstalk between these different inputs and outputs. And so we can see clearly in terms of ligands, we can map certain ligands to certain outputs and certain common pathways to certain outputs. Using this method and using a coarse-grained approach towards model building, we built the first set of models which defined for multiplicity of inputs into your cell, into your physiology for inflammation, what are the types of outputs that you can see in terms of inflammatory factors. In fact, we showed that with 10 regulatory models, you can describe the complete gamut of relationships that the macrophages show in response to stimuli. This is the type of work that will allow you to start thinking about building network models of your cellular responses to stimuli. And so we built a very complex network. We made a lot of predictions. We experimentally validated those predictions. This is the type of modeling that helps you build networks. In the second approach, in using simulation methods, in fact, a lot of engineering goes into these kinds of approaches. I have a number of engineering students who work on this problem of building linear dynamic input-output models. We derive networks from these statistically significant correlations from these linear input-output models from matrix approaches. And we built the very first signaling crosstalk network that connected various types of signaling molecules towards introducing outputs. The most important part of this approach deals with the dynamical networks. In addition to static networks, you can build complex dynamical networks. I want to walk you through information theoretic approaches. We have now started working on post-Shannon information theoretic models. In fact, you can build a combination of probabilistic models towards reconstruction of networks from data. In using mutual information, you want to maximize the mutual information, and this is exactly the approach that we take. I'll walk you through 5 slides that will give you the mutual information approach. In the same problem, we took the protein cytokine network reconstruction problem. We built first the histograms and probability mass functions, and then for each of these inputs and outputs, and then we built the cross probability functions, joint probability distributions for each of these, and then we use this approach towards building threshold coefficients for this crosstalk between these inputs and outputs, and then we use this to use an algorithm to look at the threshold towards reconstructing networks using a kernel density optimization approach. And using this approach, we can build very complex networks, and these are the type of networks that after finding that will help us in terms of redesigning experiments and redesigning therapeutic targets for experiments in real physiology. So I would like to, many examples of this go on, and we have done many applications of these approaches towards reconstructing networks. I want to walk you through now, in this part of the talk, I want to walk you through how do we look at phenotypes, build models of phenotypes. So we looked at 4 different types of macrophages. We looked at peritoneal macrophages, thioglycolate macrophages, bone marrow macrophages, and cancer cell line macrophages. We asked the question, how do these different macrophages behave in your physiology? And we used mouse models as examples. This is, in fact, one of the finest ways of getting quantitative data on metabolites in macrophages. Using this large grant, we built a multiplicity of mass spectrometric methods which very precisely quantitatively measure thousands and thousands of metabolites, in this case lipid species, very precisely. We use informatics strategies, statistics and modeling strategies to use this to reconstruct networks involved in these. And this is the very first example of a mammalian metabolic network involving a few thousand metabolites, lipid metabolites. In fact, Dr. Wang was one of the collaborators in this project, and we built this really very first network of macrophage lipid metabolism. I'll tell you the physiological consequence of this in the next few slides. So we wanted to convert this into a quantitative model. The reason to build a quantitative model from here is I'll tell you the consequence of this has relevance for pathology. If you look at the different lipids, this is a textbook picture of what happens in a fatty acid, what happens in lipid metabolism. If you take Streyer's textbook, biochemistry textbook, this is what you see. But what we can do with technology and engineering approaches is to identify quantitatively how many of these species undergo changes under inflammatory conditions. In fact, the things that you take, I know many of you take aspirin and ibuprofen. What this aspirin and ibuprofen does is to bind to specific enzymes which trigger these inflammatory molecules. In fact, eicosanoids, which are the ones on the left, are the molecules which are activated upon inflammation. In fact, the 2 pathways that are built in consequence based on these measurements trigger prostaglandins which mediate all pain and all inflammation. And in fact, the first enzyme is called cyclooxygenase, known as COX1s, and the second one is COX2. In fact, aspirin binds to COX1 and COX2 inhibitors. And we asked the question, how can you really quantitatively model these pathways so as to ask the question, therapeutically, what would be the right way to intervene with COX1 and COX2? We built quantitative kinetic models. In fact, many of you are familiar with kinetic models. We have published a number of papers on kinetic modeling approaches, and we used the two pathways to build quantitative reaction models. These are reaction kinetic linear mass action kinetic models. And in both these pathways, when we build the models, we show that we know how exactly the flux of these different metabolites go through in each of these different pathways. From these flux analysis, we can now ask the question, what type of inhibitor works best for what type of enzyme? If you look at these two enzymes, COX-1 and COX-2, we divided them into two different pools of fluxes. In fact, we can measure them using isotopic tracers, isotopically labeled tracers. And if you look at the measurements, we show that these two different enzymes show different characteristics and physiology in terms of inhibition. I'll give you an example of this. In fact, PGD2, which is prostaglandin D2, which is one of the enzymes formed as a consequence of COX-1 and COX-2 action, has no preference for COX-1 or COX-2. Both enzymes act equally well. PGE2 is coupled preferentially to COX-2. So if you want to reduce prostaglandin E2 level, you want to inhibit COX-2. And PGH2, which produces thromboxane, is again inhibited only by COX-1. So therefore, we now are able to tease out quantitatively what type of inhibition happens in pharmacological conditions. In fact, if you look at implications of this for drug design, in the case of PGD2, you have a non-selective inhibitor because any inhibitor will work equally well in terms of inhibition. If you take PGE2, you need a COX-2 specific inhibitor. If you take thromboxane, you need a COX-1 specific inhibitor. So these types of quantitative models allow you now to start thinking about inhibition therapeutic interventions in very precise ways. In the last two examples of my talk, I want to walk you through more physiological examples and give you insights into how we can build these models to understand pathology and physiology. The first one deals with a very, very exciting system we looked at, which deals with adaptation to hypoxia. Many of you have seen, in fact, papers which have come recently where they sequenced people who live on top of Himalayas and top of Mount Andes so that you can look at the differences between people who are high altitude adapted and normal human beings. That's adaptation to normal long-term adaptation. Hypoxia is also very relevant for multiplicity of diseases that you know of. In fact, heart attack, which is one of the most important, one of the common themes, hypoxia is one of the major causes for tissue damage, organ damage. You have, stroke, which is another example, and of course, cancer with solid tumors, which survive despite hypoxic conditions. So hypoxia plays a dominant role in all these diseases. So we asked the question, what are the models and mechanisms that produce hypoxia tolerance, and can we find interventions for hypoxia tolerance? The model system we used in this case were flies. Flies are excellent model systems because they have life cycles which are well contained, small, and flies can adapt all the way from 21% hypoxia to 4% hypoxia over generations. So what surprised us dramatically was the following. Let me show you in just a minute. They have short generation time. They are 75% human disease genes are orthologous to flies, so we can understand similarities between those. We use many genetic tools, and tissue exposure to ambient oxygen can be done through the tracheal system, so you can put it in an oxygen chamber, and you can look at the changes in hypoxia. And in fact, I came across this beautiful experiment done about 2 decades ago, where Gabby Haddad, a collaborator of mine, he started with flies and he reduced oxygen over a period of generations, and after 18 generations or so, he was able to get them down to 4% oxygen levels. Under 4% oxygen levels, these flies reproduced and did well and survived, reproduced. They were small metabolically, they were small in size, but metabolically they were active and they functioned well. So he did another very beautiful experiment. So he took the flies, which were in 4% hypoxia adapted flies, put them back into room air, let them go through many generations and come back to normal. And then he took the normal flies and these adapted flies, which went back into room air, put them back into 4% oxygen. Those which had 4% oxygen survived. Those which were grown from normal room air, of course, died. So this clearly says there is something going on in these few generations which leads to adaptation and genomic changes, which gives rise to clearly, modeling a system which is different in hypoxia tolerance. So we wanted to understand a model of what happens in this case. So we used a combination of, in fact, generation 18 was where the change happened in the phenotype. We wanted to look at this very closely, so we sequenced many generations of these flies which are tolerant to hypoxia in collaboration with Haddad's lab. And we did 200, different pool of 200 genotypes per sample. And then we looked at this large-scale sequencing to understand genomic differences between normal flies and hypoxia-tolerant flies. When we looked at that, we identified many, many SNPs and polymorphisms and indels, insertions and deletions, and using a number of statistical techniques which are well known. And we then showed that there are, very importantly, there are 2 developmental or 3 developmental pathways that are very important, one of which is Wnt pathway, which is a developmental pathway, which is the main pathway that appears to confer hypoxia tolerance in these species. So in order to test this further, is it really true that you adapt due to this Wnt pathway changes and polymorphisms? We did the following experiments. In order to answer these questions, is Wnt signaling involved in adaptation to hypoxia? I'll tell you the relevance to physiology in a few minutes for cancer physiology. We asked the question, can we look at gene expression changes? Can we see if we can force the fly, which is hypoxia tolerant to non-tolerant, to add these genes and activate these genes and see if it is tolerant? This is the experiment we wanted to do. So we went to first the gene expression changes between normal flies, hypoxia flies, and hypoxia and room-air flies, and we looked at what are the different changes that are associated in genes with these 3 different types of flies. And again, once again, we showed that the most important pathways that came up was this one in which you have the red arrow, which is the Wnt developmental pathway. My graduate student, Meryl Gershton, she took every gene which is activatable in this Wnt developmental pathway, and then these are all the starred genes which are highlighted. And she went into a real system experimentally. She overexpressed or knocked out each gene of interest in a specific tissue in the fly using a GAL4 UAS system, which is a well-adapted system. And then she showed that can we look at hypoxia tolerance and growth of these flies at 5% oxygen. And so we want to evaluate whether this pathway is involved. In fact, she did the experimental protocol where she inoculated the culture, she harvested the virgins, she set up crosses, she flipped the adults to move to 5% oxygen, she counted an enclosed and non-enclosed pupae. When she did this, she showed the following dramatically interesting result. She looked at 3 tissues, neuron and hemocytes, which are like the immune system cells, and heart. When she drove all of this in every single case, she showed overexpressing the Wnt canonical activators in neurons protects against hypoxia in normal flies. And she showed in hemocytes, again, in each of these tissue types, she showed that you can induce hypoxia tolerance in these cases. Think about the implications for cancer. If cancer is the flip side of this, in fact, cancer is solid tumors or hypoxia resistant, if you can now instead of activating the Wnt pathway, if you can block the Wnt pathway, you can really think about looking at going after solid tumors. We have a collaboration where we are starting to work on investigating therapeutic investigations of solid tumors with Wnt inhibitors. I want to show you the last part of my talk. I want to talk to you about systems medicine challenges and how we deal with in terms of building model systems that can deal with it. The first question that comes about in normal pathology is what is a system-wide response of physiology to pathology? In fact, most of the drugs and pharmacological interventions are focused on a very narrow or localized tissue. However, we all know that it's a system-wide response of physiology to pathological conditions. You only need to look at insulin resistance to look at the cause of blindness and diabetes and everything that goes, obesity and everything that goes with it. We want to look at cell and tissue response. You want to look at biochemical response. You want to integrate, as Peter Hunter's slide pointed out, you want to integrate across these multiple levels. How can we take the system-wide response to individualized medicine? I want to tell you a little bit about this in terms of building models. When we build these models, is it good for a class of population? Is it good for everyone? Are there molecular, mechanistic, and phenotypic network features that can categorize response of drugs? In fact, it's the same response good for everyone, the same drug response common to everyone. This is a question that we are going to answer in the following next few minutes. What interventions can alleviate pathology without causing side effects? In fact, if you go to FDA, this is the question that's posed in every single meeting. How can we avoid all the side effects associated with these drugs? In fact, majority of the drugs which are cautioned or banned are not banned because they are not effective. They're effective and more than effective in causing other side effects. What are the long-term consequences of pathology to individual physiological mechanisms? And finally, what type of models can predict long-term consequences of specific treatments to physiological mechanisms? So I want to walk you through an example from my lab where we looked at defining disease networks using the functional modules in the context of skeletal muscle and insulin resistance in responders and non-responders. I'll go through these examples. We have a number of publications, so I'll not go through every single detail. This work was largely done by two graduate students in my lab in collaboration with a postdoc and a graduate student. And we had a collaboration with a diabetologist who had a lot of human patient samples and human patient data. The basic study was focused on insulin resistance, which is, one in every, 5 or 6 Americans has some level of insulin resistance. The peripheral tissues, adipose, skeletal muscle, liver are all sensitive to hormone insulin. Primary defect leads to adult blindness, kidney failure, non-traumatic amputations. 24 million people have diabetes, which is an extreme case of insulin resistance, and 24% are undiagnosed. The mechanisms are not well understood. There are a whole lot of GWAS studies and genome-wide association studies which have shed more confusion to the problem than any light on the problem. How treatments agree and differ in their effects is not known. What biochemical pathways are involved is not known. In fact, our approach was to take these human tissues. We looked at many drugs. We looked at thiazolidinediones, and these are the drugs which are given to a lot of people. And in fact, these drugs act in a very, very well-defined manner. They bind to a transcription factor called PPR gamma, and they change genes involved in metabolism, genes involved in inflammation, genes involved in muscle activity, and so on and so forth. And so as a consequence, these have been administered as drugs for a number of years. And in fact, the cellular mechanisms are not known, and a fraction of the people, 30% of the subjects, do not respond to this treatment, so you don't know whether somebody is responding or not. So we wanted to use this approach. Our study was based on 72 human subjects, ranging from insulin sensitive to insulin resistant. We used samples from muscle and samples from fat tissue, adipose tissue from humans, and the clinical characteristics, phenotypes we looked at included RD values, glucose disposal rates, body mass index, age, glucose, insulin, hemoglobin, etc., a large number of clinical phenotypes. And the tissues we selected were vastus lateralis, which is skeletal muscle, and adipose tissue, which is subcutaneous abdominal tissue. The conditions we used were, we did two things. We made the muscle look like as if it was fed and not fed, because you can subtract the effect of feeding as a consequence, as a causation of insulin. And second, we looked at treatment, 3 months of treatment, and we looked at response of these tissues before treatment and after treatment. And so this is a systems level analysis of the disease. And then we looked at patient groups. We categorized patients purely in terms of the observed phenotypic measurements. And so if you look at patients, these are normal, the ones encircled, or the normal patients who have glucose disposal rate, which is in the right range of over 8, and who don't change with treatment. They are controls. You treat them, nothing happens. They continue to remain robust and healthy with the same glucose disposal rate. The one on the left, who are glucose disposal, who are in the RD values less than 8, or insulin resistant people. This is insulin resistant. Those on the top are the ones which improve after treatment, because their RD fraction, the RD fraction is defined as after treatment minus before treatment divided by before treatment. And so you can see that they are the ones who improve, and they show improvement in terms of their glucose disposal. These are the ones who don't improve at all. The ones in yellow are the ones who continue to maintain insulin resistance, and the others are in between. So we developed a large number of methods for analysis of vast amounts of data from these human tissue samples. We did measurements of genes, we did measurements of metabolites. I'm going to only tell you about a small piece of the story. A lot of this is well published before. So we developed a method where we used a hypothesis testing approach towards variance modeling of this data so that we can have very precise quantitative measures of what genes contribute to these pathologies. So we looked at variance model estimation, and we exploited the intrinsic expression noise in the system so that we can really ask the question, what is significant, what is not significant. We also built models for asking what is functionally relevant. Many times you get all these, when you do a standard gene expression analysis, you get all these thousands of genes. You put them into some categories. You don't know whether it's significant or not. We used sophisticated methods, developed methods to look at whether they're significant or not. I won't go into detail. So let me focus on 2 tissues and tell you the consequence of doing this. We looked at skeletal muscle tissues. The 3 things which stood out in this disease from the models we developed are 1, muscle adiposity, 2, metabolism, and 3, myogenesis. Now, the second part of it, metabolism, is well known, which is the reason why people are given this drug, because they improve in their metabolism. At least the people who respond improve in metabolism. In muscle adiposity, this was a really interesting revelation. I'll walk you through the next few slides. In fact, this made Pfizer, which collaborated with us and funded us for this project, sit up and look at their drug and ask the question, what is going on here? So what we found out was that, not surprisingly, in patients who are insulin resistant, there's increased markers of fat in your muscle. So there are a lot of fat, essentially your muscle gets morphed into fat and muscle tissue. So this is not surprising, this is known from before. In fact, all of the markers that are involved in fat are present in this muscle. In fact, but what is dramatically interesting was the following. If you monitor this slide here, the one in blue is the one, these are normal patients. This is insulin, the slide I'm showing you so far is pre-treatment with drugs. This is after clamp, which means you're fasting and feeding. When you fast and feed, you're supposed to improve in your ability to, you're supposed to not change anything in your fat markers in your muscle tissue. If you look at normal patients, who are in blue, there is no change. If you look at insulin-resistant patients who are in the 3 different colors, they increase in the fat markers in insulin-resistant tissue. It doesn't change very much with eating or not eating. You continue to maintain this fat. So this is a long-term phenomenon. When you treat with the drug, I would like you to watch this carefully. When you treat with the drug, in normal people in the blue, nothing happens. In the case of patients who are responders, which is in the light color, the fat markers go up, which means if you're supposed to respond to this treatment, instead you're increasing your fat content in your treatment. In fact, this is quite dramatically surprising. One of the early results, now there are many papers which show this in various model systems which look at it in different drugs. So you have increased adiposity associated with treatment in responders, not even in non-responders, but in responders. So even though you respond to the treatment in terms of metabolically, you're increasing your fat content. We showed this in a number of cases of fat markers. This is not the only example. Our models extended to a large number of fat markers, which included those which form lipid droplets in your physiology, in your muscle, which are involved in transport of fat, which are involved in fatty acid biosynthesis. So these, essentially the point I'm making here is these drugs amplify your fat markers in your muscle. The responders have a more pronounced response as opposed to a less pronounced response. We looked at the metabolism in the muscle. Metabolism is exactly what it is supposed to do. In fact, these drugs improve metabolism. This is before treatment. When you eat, for example, your ability to digest goes up. When you, for example, when you go out and have a snack, you increase your ability to metabolize, which is what you see in blue in normal patients. In insulin-resistant patients, it is less pronounced. You don't increase as much, which is why you're not able to metabolize. When you do a treatment, in the case of patients who are responders, do well. Patients who are non-responders don't do well. So our models now were able to start identifying a panel of markers. These markers are now able to ask the question, are you a responder or a non-responder to treatment with these drugs? So we started building a number of markers of insulin resistance. In fact, we have now panels which are used routinely to ask the question whether or not you respond to treatment or not respond to treatment. Let me talk about fat tissue. This is really, again, a very interesting example of fat tissue. There are two factors which help with, which are the cause of insulin resistance here, which are caused by insulin resistance. One is inflammation. The second is lipid metabolism. The inflammation, in fact, we look at in our model, we look at inflammation as being the dominant problem with fat tissue. Essentially, your fat is on fire. So you want to look at how do you deal with inflammation in your fat. So I want you to look at this carefully. These are all the inflammatory pathways we reconstructed as models from our measurements. The red ones are the ones which are inflammatory markers that go up in insulin-resistant patients. Everything in red is the one that goes up. I want you to watch the amount of red. In the next slide, I'll show you what goes down after treatment in responders. So in responders after treatment, you have substantial decrease. Let me go back one. If you go back one, this is in before treatment. This is after treatment in responders. In non-responders, not much happens. In fact, all of these continue to be high inflammatory markers. So at least we now have the ability to ask the question, who are responders to inflammation and non-responders to inflammation at the systems level? I'm going to give you a more focused example of this. If you look at antigen processing, which is one of the inflammatory response pathways, this is for insulin-resistant patients, and this is responders after treatment. One more example of leukocyte migration. This is before treatment in insulin-resistant patients. This is after treatment. So in fact, we show in this case responders deal well with this drug in terms of ameliorating inflammatory pathways. When you look at this is another new finding from our laboratory this is the first time we showed that in fact a branched chain amino acid pathway involved in metabolism is associated with insulin resistance. Now there are a number of pharmaceutical companies starting to design specific drug molecules and inhibitors which focus on these pathways. In fact, we showed the correlation of all these genes in these branched chain amino acid pathways. We look at the TCA cycle, the amino acid branched chain pathways. This is leucine, isoleucine, valine metabolism, and then oxidative phosphorylation. When we published this paper, we then identified specific markers which are predictors of TZD-mediated insulin sensitization. These markers are now being explored by pharma to identify specifically who are responders and who are not responders. I want to conclude this talk. When we published this paper, we were asked to show how about these different drugs? Do they act by a same mechanism? Do they have different mechanisms? Do they do the same thing? So we couldn't do it in humans because we didn't have the statistical power to build our models. So we then used an effective rat system. We used to ask the question, how do PPR gamma ligands differ in their effects, and are the mechanisms the same? We used a Zucker fatty rat, which is homozygous, obese, insulin resistant, and heterozygous lean control. We treated them 3 weeks with each of these ligands. We selectively looked at the differences between each of these ligands. In every one of these mechanisms we identified, we harvested the liver tissue, the muscle tissue, and the adipose tissue. When we looked at this, I want to summarize for you the result. Don't want to go through each one of those results. We identified the following new insights we found. In the adipose tissue, the ligand-induced insulin sensitization is correlated with anti-inflammatory effect, induction of the branched chain amino acid pathway. There is no difference there is only selective difference between these different ligands. In skeletal muscle, these ligands show increased expression of adipocyte-centric and slow twitch fiber muscles, not the fast twitch muscles. And then in liver, the PPR gamma ligands reverse the increased fatty acid synthesis. So even though we don't show it in humans, we're able to show with rat model systems how we can tease out differential effect of these different drug molecules. So I hope I've tried to convince you how you can use data-driven models, quantitative models, towards understanding physiological responses to physiology and pathology. I want to conclude the last talk with the summary of various other biological systems and models and some examples from my laboratory. We worked on the genome as a system. We have published a number of papers where we have looked at the whole-scale genome, whole-scale models of genome systems. We looked at a cell as a system. In fact, we showed using macrophage cell as an example how we can build systems-level models of cells, how we can build quantitative input-output phenotype relationships, how we can not only build data-driven models but equation-driven models. In fact, we now have not only looked at deterministic models, but you look at stochastic models where concentrations of specific species are very low and they are distributed over different parts of the cell. We have worked on the biological processes system. In fact, we looked at the lipidome, we looked at the changes in atherosclerotic effects on tissues in mice, in murine systems, and we looked at models, how to build quantitative models of these systems. We looked at the biochemical pathways. This is for many years, we've been looking at biochemical pathways as model systems, looked at qualitative and quantitative effects. In fact, experimentally, we have shown how a G protein coupled receptor pathway couples to different proteins and causes a phenotypic consequence at the end. We have quantitative results which are validated by using fluorescent marker type experiments. We looked at functional modules as systems. In fact, we built a specific functional module common to hundreds of drug molecules, which is the GTPase module. We asked the question, if you look at GTPase, it's involved in everything from your vision, which happens in milliseconds to seconds time scales, all the way up to developmental processes, which happens to weeks to months. And so we asked the question, how does this module adapt itself to these multiple time scales? We looked at variation in concentrations across this, each of these species in this module and how it is modulated in physiology. We built quantitative models that help us understand how these mechanisms change depending on the context. We built a physiological function as a system. We, in fact, showed the very first example of building the ciliary apparatus. We compared the different species to understand how does the cilia work. Cilia are the motile cells, and motile cells have cilia, and how do these work? For example, sperm cells have cilia. We showed what is the ciliary machinery that is common to everything from fly to human. We then worked on organ as a system. We are now working with cardiomyogenic regeneration in stem cells, and we now have built a complete model of skeletal muscle which can be used quantitatively to model skeletal muscle 2 pathologies, one Duchenne muscular dystrophy and the other ALS, which is also known as the Lou Gehrig syndrome. So these are the examples of models that we build as a consequence of doing this. And I just walked you through showing you the disease as a system. We used insulin resistance as an example to show you how we can build data-driven and quantitative models. I want to conclude by telling you that all of these approaches are possible by the 3 pillars of engineering. Measurement, which is really based on new technologies. There's going to be, in the next few generations, we are going to see incredibly new technologies, especially mesoscopic scale imaging, where you can image molecules in vivo at different scales, molecules and cells, through processes as a function of time. This is going to revolutionize the way we can think about biological processes. And how do we integrate this with the molecular level profiling and whole physiology profiling is a challenge that we are going to come up with. This is the analysis problem. How do we build multi-scale models? In fact, Peter Hunter talked about it. I showed you the Hunter slide. The Human Physiome Project is essentially thinking about how can we build anatomical models? How can we then put flesh on the anatomical models using a combination of all the molecules and cells that constitute this physiological system, and how can we make this quantitatively valuable in terms of equation-driven models. This is an approach where my lab has been involved in for a number of years. We are really working on the interface between data-driven and equation-driven modeling. Third approach is building experimental model systems. This is, in fact, a really interesting question. Traditionally, if you go to a pharmaceutical company, they use mice as models. Now it's becoming increasingly clear only in certain cases you can use mice as models. In some cases, you need primate models or even humans as models. In fact, we did an exhaustive comparison between different models for macrophages, for mouse, for rat, for human, and these comparisons showed dramatically interesting differences between these different model systems. We did this in the case of insulin resistance. We show how the drugs that we deal with routinely, which are tested on mice, have different phenotypes compared to what happens in humans. So defining model systems is going to be an interesting challenge for the coming times. And the last part, which I did not cover in this talk well, is the design issue. We don't understand all the principles of living systems in terms of being able to engineer and design. Many of you know that synthetic biology is now becoming a catchword in biology. I mean, it's an oxymoron because we really don't know the principle set. We are really using nature to do the design and synthesis. But we will see down the road ability to understand principles and reconstruct. In fact, examples with induced pluripotent stem cells are one first step towards this process where we are designing and building model systems that will help us. So I hope I've convinced you with this talk that engineering has a big role to play in terms of biology and medicine, especially in building absolutely quantitative models that will help us build genotype-phenotype relationships. This is the quest of where my lab is. I'll be happy to get your input and answer any questions that I can. Thank you for your attention.
Excellent talk. So this is actually by far the most comprehensive multi-scale modeling for system biology and bioinformatics I have seen. So right now it's time for questions. Do we have any questions for Professor Subramaniam?
Hi, thank you very much for a very wonderful talk. I'm Grace Peng from the NIH, National Institute of Biomedical Imaging and Bioengineering. I wonder if you can comment on how your systems biology models can give us a taste for the future and integrating it with quantitative systems pharmacology models and the whole drug development pipeline and possibly virtual clinical trials.
Yeah, thank you for this really wonderful question. As you know that NIGMS has spearheaded this white paper on quantitative systems pharmacology, of which I'm one of the authors. And so the way systems biology integrates with this is exemplified by the last example I showed you. We are not going to ever get a drug for an individual person. So we are never going to find one drug for one person. However, when you have a class of molecules, you ask the question, who are responders, who are non-responders for treatment for these drugs? So this is the first type of approach where you can ask quantitative systems approaches towards pharmacological interventions. Ask what interventions are meaningful. That's the first approach. The second second answer that comes up is that we don't deal with the drug as interfering with a given pathway. I'll give you a very, very nice example. Celebrex, for example, we have a caution on use of Celebrex in certain patients because even though it's a fantastic COX inhibitor, it does more than being just a COX inhibitor. So systems level approaches will ask the question, what are the therapeutic interventions? What additionally happens to the pathways that are being interrogated by these drugs? So the ability to look at a system as a whole, system response as a whole. In fact, FDA is starting to think about it in terms of putting a panel of systems level responses that should be routine for every drug that we use so that we can start thinking about systems level understanding of pharmacological interventions. So this is what we proposed in our QSP paper, quantitative systems pharmacology paper. We want to be thinking about building quantitative models at systems level to look at responses not of individual molecules or individual pathways, but pathways that are common to multiple physiological functions. I hope I answered your question.
Donald from Dallas. Okay, this is a very nice talk. Really stimulating people thinking about how should we do the science. Okay, in general. So for me, I'm sitting there. So first I start with very specific questions. So when you see this, when you treat these people with insulin resistance, so you see the responder, no responders, they have drugs. So then you show them, each system, their molecular pathway or something different, right? So here, how you talk about individualized medicine for each individual person. Does you see their signaling pathway is consistent changing, for example, metabolic pathway, right? So for each individual subject, 2 responders. There may be hypertension, maybe another person have different drug, whatever the conditions. So their signaling pathway may be influenced by other things. So you may be able to see the individual difference. That's my first question.
I'd be happy to answer this question. So there are two answers or 3 different things which you addressed. The first question you addressed is, do we have certain canonical common pathways or not? That's the question that you're posing.
Yes.
Clearly, if we did not have canonical pathways, we wouldn't have similar responses to the same condition.
Yeah.
For example, we wouldn't have the same response to a virus, or we wouldn't have the same, very similar overall average response to certain conditions.
Yeah.
So we do have canonical pathways. Having said that, there are dynamical changes because not everyone deals with the same way. So the question that comes up is, is each individual sufficiently different to form a very difficult collection of number of pathways that makes it very difficult to interrogate? Or do a class of individuals based on their genotype come up with, and epigenomics, come up with a certain class of pathways which can be dealt with? We believe, based on the evidence that's now being accumulated in systems medicine and biology, that's the case where we have certain class of responses coming up. In fact, in my lab, we have done a lot of single cell experiments where we have shown that if you take a large collection of single cell experiments, thousands of experiments, you can categorize them into a few categories in a coarse-grained way, which show similar responses. So that's the second part of the question. The third part of your question, which is really, really interesting, is when you're doing perturbations and interrogations, is there a dose dependence? Is there a time dependence? This also answers the previous question. We cannot do it without having a quantitative approach towards systems biology. So in order to, even if you know a pathway, you want to know at what point you want to interrogate or intervene in that pathway. And this is really an unsolved problem unless we have sufficient number of patients, sufficient collection of statistical, power to do these kinds of
You will be able to see a pattern. Right now we still treat the system as a homogeneous system, responder versus non-responders.
Correct.
So following this question probably is more general thinking here, it's kind of like, when, as a scientist, we're all, optimistic, right? We're dealing with all this complexity, layers of the different time scales, spatial scales. So when you put all these complicated systems together, they are interacting with each other. So the interaction could be linear, nonlinear, dynamics, all different levels. So I don't know, let's looking for the 20, 15 years down the road, where we hit that wall. From system, biology point of view, so from your perspective, for the young scientist moving forward, okay, so where you see that wall will, every science move to somewhere, they have to stop and then go into another turn, right? Thanks.
No, these are very interesting questions. So obviously, there is modularity in physiology. If there's no modularity in physiology, we cannot approach any problem. The fact there's conservation in function from across species, the fact there's conservation in pathology across humans tells you there is some degree of modularity. There is a variance around that modularity. Disease is not a single point. Disease is a distribution, clearly. Normal is a distribution. So the question really you're asking is that do we have sufficient modularity to approach these modules in meaningful ways, or do we have to always think about trying to integrate it in such complex manner that we are never going to get a handle on this? So the answer to that is we believe we can design this is an engineering problem. If you take an automobile as a complex system or an aircraft as a complex system with unconnected modules, you're never going to be able to design anything. So your design principles have to be based on understanding this modularity in living systems. This is what systems medicine tries to do. In fact, we want to understand modularity. I want to tell you just one more thing about this. In fact, we are starting a very ambitious program in collaboration with Children's Hospital where we are starting to look at in fact, 85% of children come to Rady Children's Hospital in San Diego. We are starting in collaboration with the head of pediatrics to start collecting human tissues longitudinally, to start collecting human pathological samples longitudinally, to monitor phenotypes longitudinally, to have a bank so that we can address exactly the question that you're raising.
Just coming back. So no microphone. Okay. I think in the future, it's something kind of like, you remember Einstein and Bohr, they have that big debate regarding the nature is a probability or is a deterministic, right? So I think the modeling we are thinking here is still a deterministic system thinking. But eventually, where we go, right? Human beings, the billions of things coming together. It's more complicated than the car.
I'm in agreement with you. In fact, the idea of deterministic is only applicable in some cases. We have statistical and probabilistic models, which are going to be very important. In fact, we insist now that everybody who learns biology needs to learn statistics and probability. That's a requirement.
It's for you. So this is actually related for research perspective. As you pointed out clearly that there are two kinds of system biology modeling. One is the data-driven, one is you call it the equation-driven. So one of the, right now, the common barrier right now in the community, as we all know, is actually data-driven. We first have to find those candidate markers, then we can continue pursue it. But finding biomarkers, the progress has been much slower than people participated. before, is still being viewed as finding a needle in a haystack. So that's why right now the common confusion in the community is it seems like doing computational-based system biology using data-driven model seems to be heavily dependent on the luck of defining the right problem, finding the marker before you even can start thinking about system biology modeling. So that's why I would like you to provide some insight on how we can address this as a community to speed up this process, because this is very fundamental, as you've shown already today, not only for biology, but now for system medicine. That's my first question, but I do want to put my second questions here.
Let me very briefly answer your first question. In fact, the problem is worse than finding a needle in a haystack, because it's like finding a hay in a haystack. So it's even worse. So it's very complex. So the advantage you have is we need to use is the following. Number one, we truly need to have sufficient statistical power to validate markers. And as I pointed out, defining the system is half the problem, or more than half the problem. So we need to define the patient population or the model system population, the phenotype that you want to measure, because if you measure 5 different phenotypes, your markers are going to be vastly different. You want to ask the question, what is the companion marker versus what is the real marker? So to do this, you need to really have controls. 90% of biology proceeds with not adequate controls. So this is really a experimental design problem.
Which is the third pillar.
Which is very clear, very important, and it's very significant. Can computation help in this? Yes. Can computation spring out a hay from a haystack? No. You need to really have sufficient experimental power to do this.
So it's still defining the right problem is the key. Okay, so then in terms of the system biology versus the synthetic biology, do you have any insight to provide the community with regard to how we can integrate those to speed up this system medicine?
You mean synthetic biology?
Yeah.
So 90% of what people call synthetic biology is changing a transcription factor in a bacteria or E. coli or yeast and seeing what happens. To me, that is really an oxymoron. You're just simply modeling something else. Then there is a J. Craig-Venter operation where you're saying, what's the minimal bacteria or minimal genome that will function as a system? Essentially, you're taking the genome and doing some kind of cut and paste operation to see what works. That's not really going to be applicable in short term for anything useful. The more interesting aspect of synthetic biology is stem cell biology, where you have induced pluripotent stem cells. If you understand the fundamental principles of how we can regulate genomic, post-genomic changes associated with specific molecules, can we drive a tissue specifically? And this really, in my opinion, is the short-term future for medicine, where stem cell therapies are starting to yield valuable output for specific tissue lineages. And I think there is unquestionably in the next decade or two, we are going to see substantial changes in this, how to do it in vivo. It's again an engineering problem, because we know how to do it in a Petri dish. We don't know how to do it in the three-dimensional scaffold in a tissue. So this is where tissue engineering becomes very important to understand what is the right three-dimensional geometry, topology, scaffold. In fact, we need to know the anatomy so that we can start building the right anatomical structure associated with this.
Oh, okay. So last one is related to some education training, because as a two-time keynote speaker for EMBC, 2004 and now 2012, I would like to report that definitely the researchers within the EMBS community has increased in the bioinformatics research. However, comparing to the large extent of bioinformatics research, we're still limited. So I see you are the founding director for the graduate program in the UCSD graduate program in bioinformatics. So I would like to ask you to share some of the insight and comments for our community. So what EMBS as a society, we can actually branding ourselves in contributing to bioinformatics system biology, and what's the kind of advice you would like to give to young scientists in our community?
I'm not sure I'm competent to provide advice, but I can share with you my experiences in dealing with this. I mean, number one, bioinformatics and systems biology are not just simply an integration of computer science, teaching some computer science courses and some biology courses, or teaching some courses. This is not the way to approach it. The way to approach it deals with how can you be intrusive in terms of bringing computer science engineering, true engineering. There's no escape for learning real fundamental engineering principles and then bringing in biology into it as a quantitative problem as opposed to a qualitative problem. In fact, we have started in our program, when we started the program, we designed courses not to be a few computer science courses and a few biology courses, but we designed these courses to be amalgamated, integrated courses. It has evolved over a period of time, and this, I think, is the future of where it's going. Now, there are a number of educators, I mean, your institution in MIT, in Stanford, in Berkeley, in San Diego, who have started embracing this paradigm of being truly multidisciplinary in approaching. I'll give you one simple example which will illustrate the point I'm making. If you're teaching, for example, biochemistry or cell biology, traditional approach deals with teaching genes and proteins and simply cataloging all the things associated with enzymes and pathways and so forth. It has no quantitative perspective attached to it. It has no informatics perspective attached to it. It has no, engineering perspective attached to it. So we are starting to build in fact, examples where we can start thinking about these as processes, these as systems, as models. How do we bring comparative analysis towards building quantitation and so forth? So this is I think, the future of where we are going. Now, engineers, I have to say something pejorative about it for a moment, with begging the pardon of the audience for a minute. A number of people, who come from physics community, who embrace biology, think that since they know physics, they can solve most biological problems. Unfortunately, the devil is in the details in biology, and if you do not really know nuts and bolts biology, you're not going to be able to treat it as an engineering problem. It's really not a physics problem, it's an engineering problem, because you really have to worry about granularity. How do you deal with multiple levels of granularity of the model to address specific answers? The same model is not applicable for everything. A model, for example, Hodgkin and Huxley model, which is fantastic, an outstanding model, you cannot explain how a single polymorphism in a potassium channel is going to change the current unless you fit it with parameters just arbitrarily. So this is really where multi-levels modeling becomes an important problem.
Okay, so questions?
I want to comment first about Shankar's inspirational role in education. I was a postdoc with Shankar, and that was way before UCSD at UIUC, and Shankar was really leading us towards the details of biology and engineering, and it was really a lasting influence on me. But I have two questions for you. As a leader of the bioinformatics computational biology, I want to ask you in terms of measurement as well as methodology. So we are coming to even more data, but what do you think is the most important one? For example, we have starting to see high C, 5C, these chromosome folding data. And in terms of methodologies, we're starting to look at not just a population average measurement, but we start to see some spatial patterns in stem cell, for example, how they are distributed and all these conditions. What do you think we should put our efforts on in terms of both measurement and modeling and methodology development?
In terms of measurement, first we need to choose the model on which we are going to make a measurement. It is not sufficient just to have large amount of data. It's necessary to have type of data that's relevant for building a model for answering a question, biological question. Majority of data generators today just generate data, independent of what problem they want to address. So this is a fundamental problem that needs to be resolved. In fact, many laboratories now are starting to take a biological problem, asking what types of measurements they have to do, omics measurements they have to do. The second issue deals with the statistical issues associated with measurement. Most people don't seem to realize that variance is a fundamental quantity of every single measurement that you make. You need to have sufficient proper statistical design to address this variance issue, and use the variance not as noise, but use it as something valuable for ascertaining what is a signal. So this is where, in fact, the EMBS is very appropriate for this, because you guys deal with this much better than any biology or bioinformatics or systems biology person does. So really asking the question, how do we deal with measurement what is the statistical error model associated with each measurement? In fact, one of the things that whenever I review a paper, I routinely ask the question, what is the error model that's been used in this high-seq or any other type of measurement that is being done? So this, I think, is an issue that needs to be resolved before we can become truly, users of data for valuable modeling.
Hi, Jeff Sosterman, Virginia. You had mentioned the Hodgkin-Huxley model. What do you think has been the biggest success story in the last 10 years in systems modeling?
I mean, there are a number of, there are at least a few drug molecules, cancer drug molecules, for example, which have come from a systems level approach. I mean, you are familiar with some of those. There is a really good, deep understanding of, for example, modeling with reference to different stem cell germ layer, creations, formation of specific tissues has come from a systems level understanding. I mean, gene expression studies and chromatin modification has given rise to specific understanding of epigenetic modifications that cause disease. We have a reasonably good understanding now of microRNA and the role of microRNA in terms of how does it deal with pathology at various levels. And so there are many, many, many such initial success stories of doing it. In the case of large-scale modeling, Peter Hunter can give you a good example in terms of design, where you yourself have done a lot of work in terms of cardiac modeling, where it's helped in terms of designing pacemakers and other kinds of devices. So there really is starting to be an impact of models and model building from multiple levels in terms of understanding and using those as tools for next generation physiology and pathology.
I think it's a great talk. You mentioned multiple examples of the system biology and bioinformatics in the normal and the pathological conditions. I have one question. The currently bioinformatics and system biology mainly focus on the genome and transcriptome, means DNA and mRNA. But however, the physiology mainly carry on by proteins. Do you think we're missing something there? And protein and protein activities, system biology a concept?
Yeah, I mean, the reason why people focus on genome and epigenome is because you can sequence fast and very easily, high throughput and very easily. It's not because they don't want to focus on the proteome or metabolome. Clearly, there is no escape for doing the complete omics on some level or the other. And now technologies like mass spectrometry are becoming much more sophisticated that in the next decade, at least in metabolomics, we are starting to do thousands and thousands of metabolites. In the case of proteins, it is not so easy, but at least in the case of specific proteins like cytokines, there are now starting to be very precise measurements which can be done. And there is an effort in Europe, which is spearheaded in Denmark, where they are starting to develop a library of human-specific antibodies and mapping the entire physiome, asking the question what proteins are present. And this hopefully will kind of lead the way towards understanding the proteome in a better light. But the complexity is very large and measurements are still not quite there. That's the reason why we are not seeing the effects of that. The second facet of your problem is even when you get those different ohms, how do you integrate them? There are different scales, different length scales, different time scales, different, functional scales, phenotype scales. So how do you integrate them? It's a very complex issue. This is really the crux of the problem that a lot of modeling people are going to deal with.
Okay, so as Jim and I talked, like any opera, the finale, right? We save the best for the last. So indeed, Professor Subramaniam gave us a wonderful talk. I consider it the best today. Let's thank Dr. Subramaniam again. Thank you very much.